log in right now, so I will go ahead and kick things off. Thanks everyone for joining the first of what will be many webinars offered by the International Oak Society. I think we said we'll probably host these quarterly, and I think we may, if we have time at the end, Roderick will probably ask for some feedback from the group if possible. People would like to share now or via email afterwards. But for the agenda today, I am just going to go through a couple of announcements for upcoming events with the International Oak Society as well as with the Global Conservation Consortium for Oak. And then I will pass it off to Dr. Valerie Pence, who will do a presentation on her work with a project that was funded by the iOS Oak Conservation and Research Fund, as well as highlighting some newer projects that they've been working on as well in her lab. And then after Valerie, we will hear from Roderick Cameron and give an overview on the collections at Grigadale Arboretum. And then we will have time after each presentation, so after Valerie's presentation, we will have time for some quick Q&A for any questions for Valerie, and the same for Roderick's presentation. And then we'll have time after both presentations to see if there are any more questions. And then I will pass it off to Roderick for the feedback and ideas moving forward for these series of webinars. So in terms of announcements, there will be a National Oak Society tour of Oregon that will be going on at the end of September. So I will share a link to that right now for folks who are just interested in learning more. And then also, iOS will be hosting Oak Open Days in Texas. So that will be a series of visits to gardens throughout Texas, as well as doing a visit to kind of the country hillside in Texas as well. So there is a link to that event for more information. And then also for the consortia purposes, there will be a cryopreservation working group meeting. That is a working group that Valerie Pence and her team help lead, and it is an opportunity to hear more about ongoing cryopreservation projects. And those meetings are hosted quarterly, and this is the sign-up sheet to that working group. So if you are interested in joining the next meeting, that will be on September 6th. Just put your information in that sign-up sheet and you will receive the calendar invite. And then finally, really exciting news, the iOS announced the 2025 conference dates. So Antonio Gonzalez Rodriguez, who will be organizing the 2025 conference in Oaxaca, Mexico, has announced the dates for the events, which will take place October 3rd through the 6th, 2025. The pre-conference tour is scheduled to begin September 26th, and the post-conference tour will end October 12th. So save the dates in your calendar now. Valerie or Roderick, did I miss anything for announcements or anything else you wanted to add?
Oh, I think that's good. Thanks very much, Amy.
All right. Well, yeah, thanks everyone so much again for joining and taking the time. Really appreciate it. So I'm very excited to now pass it off to Valerie, who will be giving the first presentation. So whenever you're ready, Valerie.
Thanks, Amy. Let me see if I can share my screen here. Are you seeing that?
Yep, looks great.
Great. Okay, well, hello, everybody. What I'm going to do today is talk a little bit about our Conservation Research Fund project, which was entitled Shoot-Tip Cryopreservation for Conserving Oak Biodiversity Ex-Situ, and also provide a couple updates. And my co-author, Jillian Ross, is or was an intern with us during the time that we had that grant from iOS. I work at the Center for Conservation and Research of Endangered Wildlife at the Cincinnati Zoo. We're called CREW, and we deal primarily with ex-situ collections of endangered plants, both in vitro and in liquid nitrogen in our cryobiobank. We focus on exceptional plants, which are species with seeds that can't survive conventional seed banking. And as most of you know, oak seeds can't survive the drying that's needed for seed banking, for preparing seeds for seed banking. So they will require alternative methods for long-term ex-situ conservation. And those methods will center around living collections, such as those that are in botanical gardens that are being coordinated by the GCC Oak Group, and then also cryo collections, which is the focus of the GCC Cryopreservation Working Group. And so I'm going to talk a little bit more today about the cryopreservation aspect of things. And as Amy mentioned, the cryo working group meeting is coming up in September. Well, what is cryopreservation? Basically, it is storing tissues in liquid nitrogen, and this is just an illustration of that process, and it's very similar for many of the procedures that are used, the protocols that are out there for cryo. But the one that's being used mostly these days is called droplet vitrification, and that's what's illustrated here for shoot tips. The shoot tips are we take just the very tip of the growing shoot, usually from an in vitro culture, and they are dissected out, about a 1 millimeter shoot tip, and then used for the cryopreservation process. The shoot tips are protected in liquid cryoprotectants of various sorts. We generally use a cryoprotectant called PVS2, which contains sucrose, glycerol, ethylene glycol, and DMSO. So it's sort of like an antifreeze solution for these tips. We also, well, after the the cryoprotectant step, the shoot tips are put onto small pieces of aluminum foil in tiny drops of the cryoprotectant, and then immersed directly into liquid nitrogen. They might stay there for an hour if you're doing an experiment and you want to test, you're just testing the procedures, or they might stay in liquid nitrogen for years if you're actually banking the tips. When you bring them out, they're thawed by immersing them in a rinsing solution, which pulls away the cryoprotectant. The cryoprotectants can be toxic if they're left on the tissues too long, and so you want to get rid of that at the end. And then the tips are put back onto a tissue culture medium and regrown into shoot cultures, which could then ultimately be grown as plants. So that's the basic process that we're looking at. I'm sorry, I'm trying to see if I can I'm sorry, that's not what I want. I'm just seeing all of your faces on the right side of my screen. I couldn't quite get rid of that, but so I might miss something that's on the right side of my screen, but we'll. The cryobanking process has been used a lot for crop species, and the USDA National Laboratory for Germplasm Resources Preservation in Fort Collins, Colorado, is banking a number of crop species using shoot tip cryopreservation, but also dormant buds. They have a collection of apples and some other woody species where they have frozen them as dormant buds. There are a number of centers around the globe that are working on crop species of various kinds internationally. And some of those, like the International Potato Center in Peru, are storing shoot tips in large numbers to bank those as a backup to their field collections. And these types of projects are banking hundreds of samples of crop species. But cryobanking of wild species is more recent as an enterprise worldwide. There are actually fewer species of wild origin in cryobanks. And some of the reasons for that is that there's just more diversity of species that people need to work with. And so trying to develop protocols for the different species has greater challenges. And oaks fall into that category. They have specific challenges, but they also have some opportunities in terms of what you can actually cryopreserve. And I think of that as an opportunity. Not every species that needs to be banked in cryo has the chance of being done in a variety of ways, and oaks have that. It's been shown that embryo axes can survive cryopreservation, somatic embryos, shoot tips, both from in vitro cultures, can survive cryopreservation. Pollen has been cryopreserved. Dormant buds really haven't been investigated too much with oaks yet, but that seems like a good possibility with some of the cold-adapted oaks. So there's a number of ways of approaching cryopreservation in oaks. But there are some challenges with oaks. Many species or stressed will produce phenolic compounds. And in culture, in tissue culture, that can be a real problem because the tissues get so stressed that they produce these compounds. The compounds can become toxic and just kill the tissues, as you can see in these pictures here. So trying to balance wounding the tissues or getting them stressed through the process of cryopreservation without having too much of the phenolic problem is a challenge with oaks. Oaks also go through episodic or seasonal growth. So tissues are only available certain times of the year, and they may have sort of an internal developmental clock that may affect their growth in vitro. So that when we want them to grow, they might not want to grow. And so there are those kind of issues as well. Also within the genus Quercus, there's a wide variety of species and adaptations. So one method might not fit all oak species. Well, our oak cryopreservation project has been going on for a number of years. But when we were working under the Oak Conservation Research Fund from the iOS, we were looking specifically to initiate multiple oak species into culture to sort of increase our collection so that we could compare more species in the future. We also wanted to evaluate shoot tip survival through cryopreservation to help improve survival. We were focusing particularly on Quercus hinklei. And we were also planning a virtual symposium and workshop to look at cryopreservation in oaks and other species. Well, during the time of our grant, we increased the number of species we had in culture quite dramatically, and that was with the help of Max Winkeljohn, who really spearheaded getting a lot of material from many of you all probably out there. He was asking for acorns, and I think he still is doing that, and growing the seedlings and initiating cultures from them. This just illustrates what our shoots look like in culture. We grow them on a woody plant medium with some cytokinin, and they are generally have been cultured at a constant temperature with a 16-hour day, 8-hour night cycle. Max in our lab is a PhD student, and he's been working particularly with media improvements to try to improve the growth of these cultures, and looking at silver thiosulfate, which can influence the oak initiation process. It seems to improve the success of oak initiation. Here in these pictures, it just shows that STS can influence the growth of just the standard maintenance cultures. And STS is interesting because it's an inhibitor of the plant growth hormone ethylene, which it's produced by plants under stress and can inhibit their growth in culture. So with some oak species, it seems like STS can help in initiation and perhaps even in growth in culture to help alleviate some of that stress. Let's see here. Well, the oak species that we targeted first for our cryopreservation studies, actually before our iOS grant, were these 4. And today I'll mention primarily Quercus virginiana, the live oak, and Quercus hinklei, which is critically endangered rare oak. And for our cryopreservation process, we followed the protocol that I showed you a little bit earlier. We take the very tiny shoot tips from the cultures, and that's really the rate limiting step, is that dissection of, dozens of these shoot tips to run an experiment. But we cryopreserve them, as I mentioned, with PVS2, which is our cryoprotectant. And we look at survival both after just the PVS2 itself, because as I said, PVS2 can be toxic if you leave it in too long, or some species might be more sensitive to it. So we take a control right after that. And then we also look, of course, at the tissues that have been treated with the cryoprotectant and then gone on and been exposed to liquid nitrogen. And after on those controls, we monitor both survival and growth. And this was some results from a paper that was published in 2021. And this was sort of where we wound up starting for our iOS grant. If you look at the table figure on the right, QV stands for Quercus virginiana, and QH stands for Quercus hinklei. And the lighter gray bars are the percent survival after just the PVS2, the control of the cryoprotectant. And the bars that are blacker are the survival after the liquid nitrogen. And you can see that we had pretty good survival after PVS2. It wasn't too toxic to these two species. But after liquid nitrogen, you did get a decrease in survival. With Quercus virginiana, we got pretty good survival, actually, for this type of an experiment. Our average was over 50%, which is not bad. Quercus hinklei, though, was down around 20% or less, and it really needed to be improved. And that's what we were trying to look at following these experiments. We did find that Quercus virginiana could be improved somewhat by recovering the tips and regrowing them in an alternating temperature. So instead of keeping them at the constant temperature of about 25, 26 degrees, we put them in an incubator where they had a similar daytime temperature, 27 degrees, and 15 degree centigrade at night. So they were getting cool nights. And you can see from these this diagram, the PVS2 controls are at the top, LN controls are at the bottom, and on the right are the ones that were in the alternating temperature. And you can see the ones in the alternating temperature did quite a bit better. But this is Quercus virginiana, the ones that were surviving somewhat well to start with. We did try these with Quercus hinklei, this experiment, and we did not see any obvious improvement with the alternating temperature recovery. And this is just to show you that with Quercus virginiana, we were able to grow the shoots out after cryo, recovered them and rooted them in soils. So we were able to recover the plants. Well, let's see, I think I missed, yeah, okay. Well, at this point, we started looking at cold hardening, and this was now with the iOS grant, and we had an intern, Jillian Ross, who worked on this for us during that time. And she decided to look not just at the temperature during recovery, but the temperature of the shoot cultures that were grown that provided the shoot tips for the experiments, the cryo experiments. And that has been called cold hardening of the shoots. So she looked at Quercus virginiana, Quercus suber, the cork oak, and Quercus hinklei. And she compared an alternating temperature, and she used a much more extreme alternating temperature, 22 degrees during the day and -1 at night. And this was taken from the literature. This has been successful with a number of woody berry species and fruit species that the USDA has worked with. And she compared that with a constant temperature. And she moved these cultures into that situation 3 weeks before they were going to be used for the dissecting out the shoot tips for cryopreservation. And she made up a table to monitor the growth, or to record the recovery of the measurements that she had on the recovered shoot tips after cryopreservation. So if you look at there's a stage 0. I don't know if I can use my pointer here. That was dead. And beyond that, there was some survival, 1, 2, 3, 4, 5. And they increased in the amount of survival and growth after cryopreservation, from 1, which was just barely any growth, to 5, where you really were getting some shoots even by about at 4 weeks. And she color-coded these, if you notice on the right-hand side there, from 0, which was white, into a dark green, which was 5. Well, this is what this looked like with Quercus virginiana again. The cold-hardened shoot tips survived and grew better than the controls, particularly for the liquid nitrogen treated ones when they were actually cold-hardened. And they looked even better than the ones that we had done before, where we just looked at the temperature in the recovery situation. Just to remind you of the color coding here, the 0 to white to green, she put these data into a table form. And if you look at where I've highlighted there, the 4-week survival of the liquid nitrogen controls, the control group on the left has very little green in it, and the cold hardened ones on the right show much more green. So there was much better growth of these tissues after cryopreservation. But again, this is Quercus virginiana. Unfortunately, with Quercus hinklei, we didn't see quite that dramatic growth. And this is just looking at the same kind of data, but in a little bit different fashion. The green shows you the percent recovery of the tissues after cryopreservation. The one on the left is the cold hardened. The one on the right is the control. 25% survived the cold hardening and 15% with the control. So that was a little bit encouraging, but this was not statistically significant. So perhaps if we had larger numbers, we could increase this, but it wasn't as dramatic as we had hoped it would be. And just to show you Quercus suber, she also tested that, and there was very little difference in the survival between the cold hardening and control. So what did that tell us? It told us that very cold nights may be slightly beneficial to Quercus hinklei as a preparation for cryopreservation, but they were not as beneficial as they were to the Quercus virginiana. So we needed to look elsewhere at other factors. But we did learn something through that, which I think was very useful. We also now have many more species in culture, including Quercus arkansana, which is another threatened species. And we did have a very successful virtual symposium workshop, which was entitled Conserving Exceptional Plants, excuse me, Cryobiotechnology and the Model of Oaks. We used oaks as a model whenever we could to show the possibilities for using cryopreservation for long-term conservation of exceptional plants. And it was very well attended, and we're planning another symposium to follow up with this in 2025. Well, just to give some brief follow-ups to the iOS grant, it really helps set the stage for our other work and other funding that we have received. And we were very excited when we last year received an IMLS National Leadership Grant for a project advancing the science of conservation cryobiotechnology, again, using oaks as a model to improve the technology supporting exceptional plant conservation. So we really think oaks are a good model, and so we're trying to pursue that. This grant is funding a 3-year study on oak culture initiation, and that may not sound directly related to cryo, but of course it is, because if you don't have the shoot cultures to take the tips from to cryopreserve them, you're not going to get very far. And so we are trying to look at the factors involved in oak culture initiation, because the whole process is not very predictable and somewhat erratic. We have a number of things in culture, but we've lost a number of things that we've tried to start in culture. And so we're trying to look to see what factors like genotype, developmental stage, the environment, genetics, what kinds of things might play into this. And we've received material this last spring from a number of partners. I think some of you are on this call, and we thank you very much for your help with this. You've all committed to giving us material the next couple of years too, so we can follow this in over the course of 3 years. We have some money in the grant to have to give to other institutions to study oak culture initiation along with us, to compare results and increase the possibilities for learning some new things. We are going to have another virtual symposium, as I said, in 2025. And here's just some a couple of photos from the early results from this material that was initiated this last spring. Some somatic embryos on the left there, popping out of a leaf of Quercus georgiana. And then on the right, Quercus olotherpensis shoot cultures. And this material, this project, or this part of the project, is being spearheaded by Dr. Megan Philpott in our lab. And then we're also very happy to be part of a project funded by BGCI and the U.S. Botanic Garden to improve cryosurvival in U.S. threatened oaks, with a focus in our lab on Quercus hinklei and Quercus arkansana. And we've been testing a number of things on Quercus hinklei. Particularly, we've started to look at Quercus arkansana, and we've had some survival with that, not very high again, but it's a start. But recently, very recently, we've had some nice results with Quercus hinklei. We've had one test where we've had 70% survival, where the cultures were grown on auger and they were a little bit older. And they were grown in this cool night situation. And an even more recent test with Quercus hinklei, where the cultures were just grown on a higher cytokinin level. And so we're very excited about that, and we're following those up. And these are just a couple of photos. Quercus arkansana on the left, which looks a little strange, but it was much more advanced than anything we had seen before. And Quercus hinklei, little shoots popping up after cryo that we were very excited to see, much better than the 10 or 20% that we're used to getting. So I think with that, I will finish. And thanks to the folks in the lab who have contributed to this work over the years, and to our funders, and particularly to the Oak Conservation Research grant that we had. We are very, very grateful. And thank you for your attention.
Wonderful. Thank you so much, Valerie, for that great presentation. Really appreciate it. We do have a question in the chat. Is there any difference on rooting before and after liquid nitrogen cryo? Any data of rooting percentage and survival rate?
Oh, that's a great question. I really don't know, because we haven't done a lot of rooting studies. We have rooted, particularly the Virginiana, but we haven't gotten some of the other shoots after cryo to develop that much. And we've been so, I think, focused on getting our cultures initiated that we really haven't gone into the rooting yet with those. But we do need to develop and study the differences in the different species, which I think will be really interesting. But I'm afraid I don't have those numbers at the moment.
Great. Thank you for that question. Any other questions or comments for Valerie? Yeah, Roderick.
Yeah, Valerie, thanks very much. That was fascinating. One thing, just to check if I understood correctly, do you have to start from an acorn? I mean, you're still back to the problem of finding acorns, or can you do this from like new growth on mature trees or from cuttings?
You can do both. And we have worked with both. And actually, our IMLS project is specifically focused on new growth from adult trees in the spring. But that is sort of a limiting factor. You get one shot a year at that kind of a thing. But in the meantime, Max has worked a lot on getting acorns and germinating them and getting the shoots from the seedlings. And actually, it seems like it's a little easier to get the seedling shoot tips to start to grow in culture, just because I think they're younger than the mature tree new growth. But yeah, we've done both actually.
Thanks.
Great, thank you. Another question in the chat. For your temperature regimens, do you take into account the temps various oak experience in their native ranges?
We haven't looked at that in as much detail. We've just been looking at Hinkley, and yeah, we use those two different regimes at the moment, and we really haven't tried to match it real closely to what it would be in its habitat. But I think that's something to really think about more closely in the future, now that we're sort of getting into this a little bit more. And we have a second incubator now, we could compare some things.
Yeah, that would be interesting. Thank you for that question. Any other questions for Valerie? Comments? All right. Well, if you think of them later on, we will have time following questions from Roderick's presentation for just any additional questions that you may have thought of later on. And I do just want to say that reiterate what Max put in the chat about if anyone has any acorns to share. He did put his email in the chat. So please get in touch with him. And thanks, Cindy, for mentioning the acorns you recently collected. That's really exciting. So yeah, thank you again so much, Valerie, for your presentation and taking the time. It was really interesting to hear about. So thank you so much. And I'm happy now to pass it on to Roderick, who will be talking more about Grigadale Arboretum. So whenever you're ready, Roderick.
Okay, let's see if I can share my screen properly.
Great.
Does that work?
Yep, looks great.
Okay, just need to see if I can access my notes. Good. Okay, so thank you, Amy. I'm actually more accustomed to guiding people around an oak collection rather than presenting it over a Zoom call. But over the next 20 or so minutes, I'd like to provide a brief introduction and, if possible, give you a small sense of what a visit to Grigadale Arboretum would be like. I will first cover the location and the conditions we have, and then give a very quick overview of its history. Next, I will discuss an aspect of management and go over the challenges we face, and also talk a little about the database we've built and how we try to share the collection online. Finally, I will present a few selected oaks from the collection and touch on some aspects of what the future presents. So the Arboretum is located on a farm in Argentina, in the province of Buenos Aires, about 25 kilometers from the coast. The latitude is 38 degrees south, which is similar to Melbourne in Australia, equivalent to Lisbon or Washington, DC in the Northern Hemisphere. We're practically at sea level. The whole park covers about 15 acres, of which about 1.5 acres is occupied by the oak collection in several sections. Our soil is slightly acidic at pH 6 and rich in nutrients due to its origin as Pampean loess, that is silt and sand blown across from the Andes. Annual average rainfall about 900 millimeters, pretty evenly spread out in the year, but summers can be dry. Temperatures range on average from about -5 degrees centigrade to 35 on our hottest days. This is meant to be USDA zone 9A. Though the frosts we get are not very severe, they can come quite late and so cause damage to trees. This map shows the distribution of Quercus and the location of Grigadale Arboretum. So one of the places in the temperate zone that is furthest away from oak habitat, what you might call the back of beyond in the oak world. The closest oak habitat, in fact, would be Quercus humboldti in Colombia, which is almost 10,000 kilometers away. So collecting oaks is not a question of walking out into nearby woods to pick up acorns or even of visiting the local nursery. The Arboretum was created by my parents. My father managed the family farm, and they lived on the homestead there. When my father retired in 1990, the land was divided among cousins. Lots were drawn from a hat, and my parents got a section with practically no buildings on it. So they built a house with its garden and park on a bare patch of land. And the location was chosen because they spotted in aerial photographs a depression that could be turned into a lake, so they would have some visual interest from the start. This is 1992, when the house was just starting to be built here. This is the depression that would later form the lake. And the only other thing there was this clump of eucalyptus, which offered some sort of protection from the southeast. And you may be able to make out the trees which had just been planted there. And this is what it looked like 27 years later, about 4 years ago. The clump of eucalyptus here was actually decimated by a tornado and then mostly removed. These are a couple of, or some of the original trees there. The basic structure of the design was a series of plantings, basically to protect the homestead from the winds, which came mainly from the south and the southeast, and to a lesser extent from the west. But at the same time remaining open to the lake, the view of the lake and the horizon. This view from the north and northeast you can show you how the homestead was protected. And this here is where the main oak collection is situated. So the oak collection actually started by my father, Duncan, and then I took it over after he died. The story goes that the interest in oaks started when my mother came back from the Chelsea Flower Show in London, and with some unusual seedlings that she had picked up at the exhibit of James Harris's Mallet Court Nursery, which you may have read about in the latest iOS journal. The focus is mainly on oak species, but he also planted some hybrids and cultivars. And my father usually planted one specimen for each taxon. His other big hobby was stamp collecting. So I guess you could say that this may have influenced his approach to planting oaks. Most of the trees come from British and European nurseries. In Argentina, you really only find 5 oaks, Quercus rubra and Quercus palustris from North America, and Quercus robur, Quercus ilex, and Quercus suber, species that are native to Spain, and so may have come over with the original Spanish colonists. My father joined the iOS early on and attended conferences, starting with the second conference in California, and some of his oaks were grown from acorns obtained at iOS seed exchanges. So here's a satellite view of the Arboretum, which I will use to give you an idea of how the oak collection is laid out. My father planted his oaks in what we call the main quercetum here, and he also planted several by the tennis court over here, close to where a Quercus suber stands, which is actually one of the first oaks planted in the park. There were also originally 2 other plantations close to the lake, which they called the oxbow, as in Oxbow Lake, and the teardrop, because of their shapes. The names turned out to be prophetic, as they were mostly wiped out by a rare flood in 2002. This image is actually from 2014, which is when we had another flood, and so you can see the problem. Only 2 of the trees from these plantings survived here. In the main quercetum, the oaks are planted about 6 or 7 meters apart. My father was a farmer, and so he did not want to take up more land that would otherwise be available for farming. And when we asked him whether the oaks were not planted a little too close together, his answer was simply, well, that's not going to be my problem. Well, it turns out he was absolutely right, of course, but already the oaks there have become a collection of trunks rather than canopies as the trees reach maturity and the canopies meet and compete for light. Great for tree hugging, but not ideal for appreciating details like leaves or acorns. So when I came to continue planting, I planted the white dots mark the trees I planted. I chose spaces further away from the house, in lines or avenues. I was able to use this space here, which is where the eucalyptus used to stand that was removed, also around the tennis court, but always planting 10 meters apart, so 10 meters between trees. I also planted along this winding access road where I managed to find some space. And here I was able to negotiate to steal some land from the farming operation and planted some Asian oaks there, and a sort of avenue around this edge here. There are several tasks involved in maintaining the collection, but in the time available here, I want to focus on the issue of thinning before mentioning some of the other challenges we face. If you're looking at the satellite images, you can see that at first, the trees here in the quercetum obviously look comfortably spaced, not so in the windbreaks, which is of course what you want them to be closer together, but soon things become quite crowded. This is 2011, 2013, so rare trees need space to develop and you want to be able to appreciate them. So some tough decisions needed to be taken. This was the case with some Quercus rubra, which had been planted, I believe, even before the oak collection was started, and we're closing in on more interesting species, including some Quercus robur cultivars, which are actually off to the right, and a Quercus filurioides and a Quercus agrifolia here. So I decided to remove the Quercus rubra, which you can see being done here, and you can see how close they were to these other more interesting trees. If you focus on the green circle here, you can see how this opened up the space, right, around the Quercus filurioides and the Quercus agrifolia. And it certainly has been worthwhile. These trees have developed quite nicely. In this case, this was an easy decision because the trees to be removed were not rare, and there were other specimens of those species planted in the park. It is harder when you would have to remove rarer species that are only represented by one specimen. So in the main quercetum, we're basically letting trees compete for space and form a rather compact forest, as it were. Some of the challenges we face when planting new trees, they obviously need watering in the first few years. That's essential to get them through summer. Floods are rare in our area, but we did have 2 once-in-a-century floods in the space of 12 years in 2002 and 2014, in both cases losing several oaks. Our ants can defoliate a young tree overnight, and if it happens often enough, that can be fatal. Hares will bite off tops of seedlings or chew around the bark, so we need protection, of course. And parrots actually like to snip off oak twigs to line their nests, which in the case of small seedlings can be a problem. But perhaps the most serious threat that has emerged is what we call bicho taladro in Spanish, which is a type of ambrosia beetle that will enter the trunk and then eat its way through the branches of a tree, causing them to weaken and eventually snap. There is no effective treatment. You can try blocking the entrance hole, injecting a pesticide or applying fertilizer, but the only solution really is to remove the tree and burn it. And unfortunately, it does spread from tree to tree. We have lost several oaks this way, and it is certainly a concern for the future. A word about the collection database. When I took over the collection, there were no proper records. Fortunately, most of the trees in the collection did have a label with just a name. But aside from that, all I had was a series of diaries where my father made some notes. So I had to reconstruct what records I could based on that. At the beginning of each year, my father would list all the oaks he had. He would also make a note whenever he received oaks or whenever he planted them out. So based on this, I was able to put a file together tracing the history of each tree. In some cases, I could find a note that indicated which nursery it had come from. In other cases, I could deduce an origin, for example, when he was likely to have obtained acorns at an Iowa seed exchange. But in many cases, the origin is unfortunately unknown. This is the Excel file I put together with a column for each year. Black means the tree disappeared from the record, so presumably dead. And basically about 50% of the accessions died. So, you know, putting it together an oak collection in these conditions was certainly not an easy task. This database was later transferred to an online database linked to our website, which we'll see in a minute. This is a collection with in a very remote location, so it is rarely visited. So I thought it would be worthwhile to create a website that would allow the collection to be accessed virtually from around the world. The website includes a catalog of all the trees with measurements and images, and also a map showing their location. So let me see if I can try to show it to you. Hopefully you see it there. So this is the website. If I go to collection, I can see a catalog which lists all the trees. If I click on a name, I'll see the specimens we have of that particular taxon. Going into each page will have a description of the tree, will have its vital statistics, the measurements of height and girth. Ideally, we've been trying to do this every 2 or 3 years, but the pandemic has caused trouble there. And then photographs, images of the tree showing the leaves, details of acorns as well, several of them, also the bark, and then the tree itself over the years in inverse chronological order. If you hover over each image, you'll see the date of it. And if you go to one of the more recent plantings, for example, Quercus bushii here, same information. And then if we flick through the photographs and go to the photographs of the tree, this is 2022, last year, and then showing the history of it, right? Going back to right when it was first planted, or when it was still quite young in 2018. Here you see with the new plantings, the protection we would have for hairs. This is actually a bit low, could have been a bit higher. And I would also use a sheet of plastic mulch, 2 meters by 2 meters, which helps by, keeping back the grass and the weeds, and also retaining humidity. So if I can go back to my slides, right. Now I want to show you just some of the more interesting oaks in the collection. Starting with the Mexican oaks, they seem to grow quite well with us, quite fast, also putting out about a meter per year. The collection does not hold many Mexican species. My father actually had 3, but I was able to add some. Quercus rugosa was planted by my father, so in the main quercetum, it's formed a fine tree. This photograph is from 2018, and nowadays it'd actually be harder to photograph because of the lack of space. As I mentioned, in the main quercetum, spacing has been an issue. Quercus rhizophila, a very attractive tree with lovely new growth. Copper colored here in the middle picture, and on one occasion it produced astounding coloring in the autumn, shown in the photo on the left. This is very unusual because this is an habitat, an evergreen or maybe semi-evergreen species. It happened in 2011, and it may have been a reaction to stress because of waterlogging or some other issue. There were already symptoms in 2010 that the tree was not happy, and you may see on the photo on the left that branches at the top are lacking leaves, and in fact it appeared to be slowly dying. So in 2014 I took the decision to prune it back very severely in the hope that it might react with new vigor. Here on the left you see it in the spring following the pruning. Unfortunately, it has actually reacted very well and now looks like a healthy and vigorous specimen. Quercus insignis, a tree that I added in the space left by the old eucalyptus block that was cleared. It is sometimes affected by frost, but is certainly surviving and displaying its attractions. Growth has been slowed down by some remedial pruning as it had poor structure, but with any luck in a few years, it will take on a nicer shape. Quercus calophylla, planted in this same area I mentioned, has grown very well. It's one of the finer oaks in the collection, obtained from Bob McCartney's Woodlanders nursery in South Carolina, and actually grown from acorns obtained in the IS seed exchange at Puebla. Interesting that Quercus calophylla with us seems to be evergreen. At least it's these pictures were taken just this month, i.e. in deepest winter for us, and showing no signs of turning color or dropping. At Hagfalls in New Zealand, Bob Berry noted that the leaves would turn red and fall in spring as the new leaves were emerging, also red. But we have not yet noticed that with ours. Last Mexican oak, this is Quercus mevi or Skinneri. It was collected by Paco Garin in Huachinango in Puebla. And he first identified it as Quercus Skinneri, then he changed his mind and thought it might be the recently described Quercus mevi. But I spoke to him recently and he's actually changed that and he thinks it really is Quercus Skinneri. In any case, a beautiful oak with these very graceful serrate leaves and stunning new growth in spring. Moving on to U.S. oaks, Quercus texana. These were planted in these ill-fated plantations near the lake I mentioned earlier, and they were the only ones that actually survived the floods in 2002. So it's interesting to see how flood-resistant these species are. They grew into this globular shape, presumably because they're exposed to wind. They turn a beautiful red in autumn. And in 2014, they once again showed how flood tolerant this species is because they were underwater for another 8 months. In the satellite image, you can see how deep in the water they were, and yet they seem to take it in their stride. What seem to be doing them in, unfortunately, is this ambrosia beetle that I mentioned earlier. We first noticed it in 2018. These branches here are actually brown. Not sure if you can make it out. The leaves are brown because the branches have snapped. And in 2019 here, this was a lot worse. And so we decided to actually remove this tree here. And this one we pruned back very severely, removing the damaged branches and hoping that it will grow back with enough vigor to throw off the infestation. More US oaks, a couple of threatened oaks. This is Quercus arkansana, which is currently listed as vulnerable by IUCN, grown very well, putting on about 1 meter a year with very good structure. Quercus oglethorpensis, which Valerie also mentioned. This is endangered. We obtained this from woodlanders and it's from wild collected seed, which is great. Interesting to see that it is marcescent with us. Lovely light brown leaves that stay on, and this is in late fall. So Quercus oglethorpensis has grown quite slowly, but Quercus minima, on the hand, has put on tremendous growth, which is remarkable because this is a knee-high shrub in natural habitat, but here it has formed a tree over 10 meters at the moment. And this growth actually made me suspect that it was not Quercus minima at all, but rather Quercus virginiana. However, it may be that simply in our benign conditions, it grows much taller than it would in the wild. Moving on to section ilex oaks, we have a few Himalayan ones that are relatively rare. Quercus locotrichophora is, of course, a beautiful species, a lovely whitish leaf undersides. Growing slowly, but still doing well. This photograph was about 5 years ago, so it's put on a bit of fair bit of growth since then. Unfortunately, I do not have the origin data for this, but I do have it for this next one, the equally attractive Quercus semicarpifolia. There was a note in my father's diaries that this came from Seaford Nursery in Northern Ireland, and I was able to contact Patrick Ford's widow. Actually, Patrick Ford died the same year as my father did, 2008. And but anyway, she was able to confirm that this was collected by Patrick in Tibet. This is a particularly photogenic species with lovely catkins and bright pink new growth and golden undersides to the leaves. Here's another special one, Quercus balut, which is very rare in collections. This was collected by Sean Haddock in 1995, and this has particularly liked our conditions and grown quite a bit larger than trees from Sean's collection in other arboreta in Europe. So this makes it the largest known specimen in cultivation, our champion, which we're quite proud of. And it's another oak with beautiful new growth in spring here on the left. Moving on to some of our cultivars. This tree died in 2016, but for many years this was a great talking point. My father planted it in 2002, or at least he dedicated it at that time to commemorate the Jubilee of Elizabeth II. My mother wrote a letter to the Queen alerting her about this, and a charming reply was received from Buckingham Palace, which of course was framed and hung up by the bar in the house. Another attractive cultivar, Quercus robur argentea marginata. It has done okay with us, though not great, perhaps because it is shaded by other larger trees nearby. A very attractive variegated foliage, and even the acorns are variegated when they're still green. Another cultivar, this is Quercus dentata pinnatifidi, very attractive. It has grown quite poorly with us, but it's still a notable tree. I had to remove a fork on this, and the new foliage that sprouted from the severed stem was particularly attractive here on the left. Also interesting was that in 2010, I found a twig with normal Quercus dentata leaves on it, which are of course quite different to those of the cultivar. You might be able to make them out here on the picture on the right, quite high up in the canopy. Quercus ceris curly head, another intriguing cultivar. I actually planted this in a flower bed. I thought it'd make a nice small tree there. The flower bed has since been removed, so it now stands as an isolated specimen, and who knows, it might actually grow quite large. A couple of hybrids to finish up on. This is Quercus warburgii. It's unusual. It's definitely half of it is Quercus rugosa, and it is suspected the other parent is Quercus robur. It has put on fast growth with us, about 1 meter a year, lovely reddish new growth. And something I've noticed is that the acorns are mottled when they are ripening, mostly greenish brown with dark brown spots like a leopard skin. I don't know if you can make it out in the photograph here. I've never actually come across this detail in descriptions of the cultivar, nor have I seen this in any other oak. Another interesting oak with us is this Quercus villemoriniana. This is actually grown from seed collected from the original Quercus villemoriniana at Arboretum National de Bar in France. So technically an F2 hybrid, and possibly crossed with another species or back crossed with one of the parents, which are Quercus dentata and Quercus petria. This produces a large amount of acorns, which immediately germinate. And at one stage, I planted a whole lot and selected 2 of the more vigorous seedlings and planted them out in a new section of the Arboretum, where they're doing extremely well. These offspring actually look more like Quercus dentata than the parent, with deeper lobing and a corky bark. So they could be a back cross with Quercus dentata, which grows nearby in the collection. The thing is, of course, that a whole load of other white oaks grow nearby, so who knows what is in the mix. In any case, a couple of promising trees. So to conclude, the thoughts for the future. Succession is always going to be a problem with a private arboretum that does not generate income. One relies on maintaining a continuous line of enthusiastic owners, or at least those who are oak enthusiasts. I personally have now moved away and live quite far from the Arboretum, but fortunately my brother who lives there and manages the farm is becoming interested in the oaks. But you have to find a reason to put in the effort and investment required to keep it going. We were able to get level 1 accreditation from ArbNet, and level 1 is probably as far as we can go given our limitations, but it allowed us to get involved in a couple of conservation projects funded by ArbNet and BGCI involving the endangered Quercus hinckley in partnership with the John Ferry Garden in Texas, and another focusing on Quercus acerifolia and Quercus arkansana in partnership with Stevens Lake Park Arboretum in Missouri. And work like that certainly justifies maintaining the collection. Now, to end with, and I fear I may have gone on too long, but just a couple of videos to show you some images of what it looks like from the air. So here you can see the plantings of these lines of trees behind protecting. This is the axis, and this here is the where I planted those Asian oaks, stealing land from agriculture. And the oak collection is located right in here. And in the next video, which I'll show in a minute, you can see, I hope, yeah, this is the oak collection here. So from the this is the Quercus filariioides I said that I was able to create more space for, but you can see in the rest here of the quercetum, really they're growing quite close together. We were able to remove these poplars around the edge, which gives us more space and allows at least these oaks to be appreciated. So with that, thank you very much for your attention. I'm happy to take any questions you might have.
Wonderful. Thanks so much, Roderick. Really appreciate that wonderful presentation. Question in the chat. Do you provide supplemental water to the oaks during periods of drought?
Yes, very much. In fact, the way my parents did it, if there was no rain, I think it was like for 2 weeks in the summer, they would try to give 50 liters to each tree. And this would involve like a huge tank, which was towed around by a tractor, and they would go around, or someone would go around and provide water. This is only necessary in summer, but certainly you would require it, otherwise the young trees would certainly die.
Great. Thank you for that question. And other questions, feel free to unmute yourself too. Yeah, sorry.
Roderick, that was lovely. I noticed from your website you focus on eucalyptus and quercus, two of my favorite genera. Is that an incomplete list, or what led you or your parents to eucalypts in particular?
Yeah, that's interesting. No, eucalypts was something I started. It's an interesting point. In Argentina, eucalypts have a bad reputation. Well, maybe they just have a bad reputation with me because they're planted, just a few species, mainly Eucalyptus globulus, and they're always planted in blocks, in these rectangular, very boring and dull blocks, or in avenues, straight lines. And so that was my idea of eucalypts. And when I discovered what the Eucalyptus genus is really like, I was fascinated by them. So I started importing seed from Australia and just growing what I could. And then in that eucalyptus block that was decimated, which was all globulus, I started planting some of the ones I'd raised. And so there are a few. There's a small number. And of course, I agree with you, it's a fascinating genus, very attractive. But at the moment, I'm not pursuing that any further.
Great, thank you for that question. Have you done any assessment of the edibility of the acorns?
Not with the acorns you produce there. Apparently, the sweetest acorns are Quercus rotundifolia, which we do not have there. I have personally tried some of those acorns from other sources, and basically, yes, it's a sort of acorn that you can chew on without spitting it out, which is not the case with most others. But no, we've not tried any edible acorns at the collection in Argentina.
Great. Interesting question. Thank you. Other questions from the group? I guess, Roderick, I have a follow-up to that. With the acorns that are produced on the trees there, do you tend to just then grow them on site and plant them out at the Arboretum, or do you distribute them, or what is your process there?
We do distribute them. We tried to, or in the past we certainly did, and trying to continue that now after the pandemic. We would organize open days where we would guide people around the collection. And this was often usually in April, so acorn time. And we would definitely distribute acorns. People would take them, germinate them at home, and being planted around the country, most likely, yes. What I mentioned about the Quercus wilmoriana, so I think there's a lot of interesting work to be done, seeing what sort of acorns we can raise. And it's actually much better for us to start with our own acorns rather than importing acorns from the Northern Hemisphere, because the acorns go through like a kind of season lag, right? Because you pick them in Northern Hemisphere fall and you bring them into late spring, summer. So I actually find that I get a lot of failures just because the acorns are not adapting to that. So it's much better to start with our own. And even if they're garden collected and hybridized, you can get some very interesting things like we did with the Quercus wilmoriana. So yes, I definitely think it is worth pursuing that.
Thank you. What software do you use, sorry, to display your collections?
The website, that's interesting. I actually came across someone who'd created a website for the street trees of Buenos Aires, and I think he'd written it himself. He was a software designer, and so I asked him, "Well, can we adapt this?" And he said, "Sure." So he adapted what he'd created for us, and then another programmer created the website. It is based on WordPress. But then the actual catalog, where we show the trees, that is this customized software that was created by, especially for this other project.
Great, thanks. Thanks for that question. Any plans to add to the collection?
Yes, that's an interesting question because I personally, as I say, I've moved to Uruguay and I'm starting my own collection here in Uruguay. But we're hoping that, especially with these projects, conservation projects, that we will be adding some threatened oaks, especially to the whatever little space is still available there.
And a question came in about why have you called the Arboretum Grigadale?
Short answer to that, the Grigadale Farm was the name of the farm in the west coast of Scotland in the island of Ardnamurchan, from where my great-great-grandfather came. So my father chose this name for the farm when the original family was divided up, a family farm was divided up, and he got his section. He chose this name, which came from the family history way back in Scotland.
Oh, very interesting. And then how does your oak collection fit in with others, public or private, in Argentina, and how do you interact with them?
Good question. There are no really public oak collections in Argentina. There's the Botanical Garden in Buenos Aires does hold a few. It actually has a Quercus germana, which is unusual, but very few. There is one other excellent collection in Argentina, which is Peter Larrague's collection, about 3 hours away from us. And then a smaller one, which was started by Italian gentleman Giuseppe di Passalacqua. He died a few years ago, and I don't believe that's really been maintained. But certainly with Peter's, we do very much in contact, and we have held joint events. So we'll have one visit at our arboretum and then a 3-hour drive, and the next day we'll have a visit there. So we've done that a couple of times, and that's worked very well. And then we also actually, some of our trees are from Peter and vice versa. So we exchange trees as well.
Okay. Wonderful. Thank you all so much for these questions. Any final questions for Roderick or Valerie from her presentation earlier? All right. Well, thank you all so much for those questions. And if you think of anything later, feel free to pass them on to us via email, and we'd be happy to answer them that way. But now I'm going to pass it off to you, Roderick, just for a couple of minutes to maybe get some feedback from the group.
Sure. Well, first of all, Amy, thank you very much for hosting this. And also thank you for the great job you're doing as chair of the Development and Outreach Committee for the IOS, which is where this project started from. Thank you also to the Morton Arboretum, was provided their Zoom service for us. But as Amy said, this is like completely new for us. We've never held a webinar. We're hoping to continue it on a quarterly basis. So it's great to see this attendance. Note that this is probably the largest gathering of iOS members outside a conference. So great to see you all here. But one thing we want to think about is what sort of content to offer in these webinars. We started off with this first one with a great presentation about a project funded by the OCNR Fund, and then sharing a collection. But it'd be great to hear your feedback on that, if you think these were good choices or other ideas of what we could show. I have other ideas. While you're thinking.
Yeah, and just feel free to unmute and shout them out or put them in the chat if you'd like. Well, maybe let's hear some of your ideas, Roderick.
Well, I think it'd be great if people could report on like seed collecting trips, or like their own trips they've done. I believe we have Tom Hudson on the call, who's done some fascinating trips in Southeast Asia. Not sure if we can persuade him, but I think that is something that would be appreciated. We could also have like a species spotlight, someone presenting on a particular species, so we can learn about more about a particular species, or people might present on species from their particular area. Those are a couple of things I thought of. So I don't know if you think those might be worth pursuing, or any others you might want to share.
It feels like pretty much anything that would be shown at an iOS conference, because most of us, or all of us, have attended the conferences, and and the Zoom format works well for that. So it seemed like there's quite a few possibilities.
Yeah, absolutely. I mean, we could sort of create a sort of like parallel conference stream, or parallel conferences which are smaller and shorter, and certainly we could even elaborate projects that would then be presented formally at the conference. So that's a great idea. We have a couple of ideas in the chat here. Tim, particularly interested in timber qualities and silviculture of many oak species, Chris. That's great. Is that something you might be able to present about, or you're hoping other people will present? Don't know if Chris is there to answer that.
I am.
Great.
Hi, Roderick.
Okay.
I met you at Tiverton a few weeks ago.
That's right. Remember well.
Yeah. No, I lead on silviculture with forest research in England, in Britain. So I'm looking for alternative productive species that we could grow commercially in our country. So at the moment we rely on two, really. If you're Petrea and Roba and red oak. If you count that, they're the only three oaks we actually grow in any sort of quantity. So I'm particularly interested in those that might have potential for British forestry.
Great. No, certainly would be good to hear from our British members. So sure, they'd be very welcome. Let's be in touch about that.
Okay.
Rickman Brown is saying he likes the idea of highlighting a connection each time, so we get a very unique experience. Great. Yeah, definitely. And Rick, if you ever wish to present about the I've forgotten the name of your Williamsburg, right? The collection there.
Colonial Williamsburg, yeah.
That's right. That would be certainly welcome. Sure. And Cyrus, yes, mixing up two different topics. I think that's important. Valerie's presentation was fascinating, as I mentioned, and I think it's great to have something with a cutting edge, new research, and also we could go, have something quite different and contrasting more on the amateur side. Yeah, I agree.
Yeah, and to follow up on that is hopefully, we can highlight the other Oak Conservation and Research Fund award recipient projects as well, similar to what Valerie presented on. So we'll hopefully have that paired up with maybe another topic for the next webinar as well. But obviously have webinars focused around all these other ideas that you're sharing. So thank you, everyone. Just a few more that I received. Ongoing restoration projects around the world. Love that idea. If you all have any specific restoration projects in mind, please let us know. And then webinar on techniques for IDing parentage of putative oak hybrids as opposed to peer species, including reasonably priced DNA analysis. Great idea. That'd be really interesting. And so I guess, Roderick, I have a question on, since we're getting some wonderful ideas, maybe some people interested in presenting themselves. If that's the case, should they just reach out to one of us if they want to present at a future webinar?
Absolutely, yeah. Do you want to put your email there? I can type mine too.
Yep, yeah. So
or
please
just contact us through the website.
Yes. So just email us if you want to present on something, and it doesn't necessarily need to be at the next webinar. Let's say the next webinar that we have scheduled, which we'll hopefully share in a follow-up email and get kind of a relative month for you all. But if you want to do something maybe early next year or whenever it works best in your schedule, we can definitely fit it in that way and work around how it works best for you. So you're not signing up to be the next presenter if you reach out, so don't worry about that. But yeah, other thoughts or ideas from the group? Either on topics or format of the webinar? Overseas acorn collections, like to know how those work, and anything propagation. Thank you, Greg. Wonderful ideas.
Okay, well, Amy, do you think we can wrap this up now? Anything else on
Yeah,
our agenda?
I think so.
Okay.
Yeah, I was taking notes from all these ideas. Thank you everyone so much again for your feedback. I will be following up with the various links that I mentioned earlier for the announcements I shared. And I will also be following up with the recording as well. And yes, we will be in touch about kind of a relative time frame for the next webinar also. So that will be sent in an email. And yeah, please reach out if you have other ideas or questions, or if you're interested in presenting in the future. But a huge thank you again to both of our presenters, Valerie and Roderick. Thank you both so much for taking the time and sharing your work with us. We really appreciate it. And thanks everyone for hanging on and joining us today. Wonderful to see you all and to have you participate. So yeah, I hope you have a wonderful rest of your day, afternoon, evening, everyone. Good to see you all. Thank you so much. Bye-bye.
Thanks, Amy. Thanks, Valerie.
Bye.